Hi, my name is Jorge Chavez, and today I'll be discussing the research paper Knockdown of WFS1, a fly homolog of Wolfram syndrome 1 in the nervous system increases susceptibility to age and stress-induced neuronal dysfunction and degeneration in Drosophila. The purpose of this research paper is to study the role that WFS1 has on normal neurological and behavioral function. WFS1 is Drosophila homolog of the WFS1 gene in humans. WFS1 encodes a protein called wolframin that is crucial for the function of the endoplasmic reticulum, also known as the ER. It regulates the amount of calcium in cells. Mutations to the WFS1 produces a faulty form of wolframin that is incapable of carrying out its normal role. The stress that this causes on the endoplasmic reticulum then leads to apoptosis of pancreatic beta cells. A loss of function of WFS1 leads to what is known as Wolfram syndrome. Wolfram syndrome results in childhood onset of diabetes and deafness. It also leads to optic atrophy, which refers to damage to the optic nerve that causes vision problems and eventual blindness. Figure 1 contains several experiments, but the main idea behind the experiments done is that WFS1 is knocked down and the results that this has on Drosophila is then observed. WFS1 was knocked down by using the GAL4 UAS system. Flies containing the GAL4 driver were crossed with flies that contain RNAi, which stands for RNA interference and is used to inhibit gene expression of WFS1. GAL4 produces transcription factors that are necessary to transcribe RNAi. RNAi operates by targeting mRNA molecules. This particular figure just shows the different target sites of RNAi along the mRNA. TripRNAi, which is shown in this figure, is a particular RNAi that targets the WFS1 gene in the mRNA. After knocking down WFS1, the locomotor skills of the Drosophila were tested using a climbing assay, and the results are shown in this figure. In a climbing assay, the flies are placed at the bottom of a test tube, and their movement up the test tube is observed. It was observed that the control group, which represents the wild-type flies, moved up the test tube much more than the WFS1 RNAi group, which represents the flies whose WFS1 was knocked down. This clear decline in the movement up the test tube for the WFS1 RNAi indicates that WFS1 plays an important role in locomotor capabilities. Figure 1e shows the changes in the nerve cell and optic lobe of flies with and without WFS1. An increase in the number of vacuoles is noticed in WFS1 RNAi group when compared to the control. Vacuoles do not belong in the nerves, and their presence indicates neurodegeneration. This shows that WFS1 prevents neurodegeneration from taking place within neurons. The experiments done in this paper allow us to draw the conclusion that WFS1 does play an important role in neuronal integrity. Knockdown of this gene leads to neuronal dysfunction and degeneration. By noticing the importance of WFS1, we deepen our understanding of the factors that regulate resilience to neurodegenerative diseases. In the future, research can be done to study other factors that may share a role in susceptibility to neurodegenerative diseases, such as Wolfram syndrome. With this combined knowledge, we could potentially use these findings to delay the onset or control the progression rate of neurodegenerative diseases like Wolfram syndrome. Thank you for listening.